Is the presentation visible to everyone?
Yes, Aditi, it's visible.
Okay. Thank you very much, Edna, for this opportunity to talk about the science of cellular combat, which is specifically we are discussing today ADCC and CDC. So, in today's presentation, what I'll discuss is brief about the immune system, understanding the mechanism of action of ADCC as well as CDC. We'll also talk about some of the bioassays of ADCC, CDC, a representation that we have developed at Venture Center, and certain points that we want to remember at the end of the session. So, first, the defense mechanism. The immune system is a complex network of cells, tissues, and organs that work together to defend the body against harmful pathogens such as viruses, bacteria, and various parasites. It is also known to play a crucial role in identifying and eliminating abnormal cells such as cancer cells. Therefore, the defense mechanism can be dependent or independent of different effector cells. So when I talk about different ways in which the body combats to the pathogens, it can be programmed cell death, it can be ROS dependent non-apoptotic cell death, it can be homotypic adhesion and lysosome mediated non-apoptotic cell death, it could be CDC driven cell death, ADCC, or it could be ADCP, where phagocytosis might occur. Now, when we are talking about several effector cells, the most important effector cells that we know are either from innate immunity, which are the rapid responses, and another one is the adaptive immunity, which is comparatively slow in response. So under innate immunity, we see NK cells, which are natural killer cells, macrophages, phagocytes, dendritic cells, neutrophils. These all come under innate immunity. While adaptive immunity, there are B cells and T cells. The B cells are known to express antibody that specifically bind to the antigens, and T helper cells expressing CD4 and CD8 can be associated for bringing about the combat. So let's talk about the very common and very effective effector cell, that is a natural killer cells. Natural killer cells are a group of innate lymphocyte cells with cytolytic activity against virus infected cells and physiologically stressed cells like tumor cells. They are typically large, granular, and bone marrow derived lymphocytes. The granule found in the cytoplasm are typically azurophilic and filled with hydrolytic and digestive enzymes. Natural killer cells bring about cytotoxic immune response via ADCC, various other effector immune responses, and there are checkpoints like PDL1 interactions, which keep the natural killer cells in its nascent form and does not bring about non-specific cell killing. So since we are talking about ADCC today, let's come back to our topic of interest. Now, when natural killer cells bring about ADCC, it is via Fc gamma receptors. The Fc gamma receptors are a group of glycoproteins found on the surface of certain immune cells, which can be B lymphocytes, NK cells, macrophages, neutrophils, eosinophils, basophils, and mast cells. They all play a crucial role in the immune system by binding to the Fc portion of the antibody. The presence of different Fc gamma receptors and their classification depends on the affinity for the IgG molecule as well as their cellular distribution. Different types of Fc gamma receptors present are Fc gamma R1, R2A, B, R2C. Very well and commonly known are Fc gamma R3A, also known as CD16A, CD16B, and FcRn, where FcRn is responsible for clearance of the antibody. And Fc gamma R2B are known to modulate the immune responses by reverse or negative control of the Fc gamma receptor activation. Different functions of Fc gamma receptors are phagocytosis, ADCC, inflammatory responses, regulation of immune responses. And as I said earlier, the inhibitory receptors like Fc gamma R2b help to modulate immune responses, immune reactions, and prevent excessive inflammation. Let's come to the first mechanism of action that we are going to discuss today, that is ADCC. It is called as antibody dependent cellular cytotoxicity. It is a mechanism of immune system where effector cells, we just talked about natural killer cells, kill target cells that are coated with specific antibodies. The key players of the ADCC are natural killer cells, macrophages, neutrophils, and eosinophils. Now let's look at the mechanism of action of ADCC. Consider this is a typical cancer cell and it has very specific antigens present on it, which are considered as the cancer marker, right? So therapeutic antibodies that are administered, those will directly go and bind to these antigens. Thus, the cancer cells get decorated with all these antibodies binding to the specific cancer marker. There are various circulating natural killer cells or effector cells that express Fc gamma receptors. They specifically identify antibody decorated cancer cell and they go and bind to the Fc portion of the antibody. Now, once this complex is formed, the natural killer cells is activated and it brings about release of various cytotoxic components that ultimately leads to cell death. So this is basically about how the ADCC happens. Now, what are important points to remember in terms of ADCC? Fc gamma A binds to clustered IgG molecules displayed on the cell surface of target cells, but does not bind to monomeric IgG circulating in the plasma. Therefore, ADCC occurs only when the target cell is coated with antibody. Free IgG in the plasma does not activate NK cells or compete effectively with cell-bound IgG for binding to Fc gamma 3A. What does this mean? This means that if the antibody is not bound to the cell surface antigen, the natural killer cells will not bind to the free IgG present in the plasma. One. Second is that only when the antibody present on the cell surface is engaged, only then the natural killer cells will go and bind to the Fc portion of the antibody, and it will not compete. The free IgG present in the plasma will neither compete to activate the NK cells nor compete with the cell bound IgG for binding to the receptor. Therefore, there are many therapeutic antibodies where they bind to free proteins present. Say, for example, adalimumab binding to the soluble TNF alpha shall not bring about the ADCC activity because the TNF alpha is not membrane bound and it is available freely inside in the plasma. While effector cell responses can mediate or significantly enhance the efficacy of therapeutic antibodies, it is not always desirable or can instead lead to occurrence of adverse side effect. This particularly is problematic if expression of target molecule is not sufficiently limited to tumor cells. What do we understand by this? That once the antibody is bound to the cancer marker or specific antigen present on the cancer cell, and if that marker is not very specific to cancer cell and it binds to many other or present in many other normal cell types, then antibody which are coating to cancer cells along with the normal cells, NK cell will not be able to differentiate between cancer cell or the normal cell. It will bring about cytotoxic effect on cancer cell as well as the normal cell. Therefore, it can cause a lot of side effects. So it is very important to have a marker which is very, very specifically expressed on the cancer cells. In addition to that, glycosylation is known to enhance ADCC activity. Therefore, the use of aglycosylated antibodies may further limit effector responses. So, glycosylation is important to enhance the ADCC, right? So, there can be two ways where there one wants to have increased ADCC function. It has to be a glycosylated antibody, and on the other hand, where we want to reduce the side effects or toxic effect of that antibody, a glycosylated antibodies are chosen so that there is no and ADCC activity happening. Edna, I would like, if there are any questions, we can take them during the chat answer.
Sure. Yeah. Aditi, no questions in the chat box. But at this point, does any participant have any specific query and would like to ask it? Maybe we can spend a few minutes on doing that. If any of you all have any queries, probably you can just raise your hand and we'll be happy to do that.
Yeah, do let me know. I'll till that time I'll continue, and we can take a break whenever there are questions, because I want this session more interactive so all of us can engage and understand the mechanism very well. So coming back to the ADCC, it has been highly recommended to identify and define the characterization of the monoclonal antibody, specifically when there is a development of biosimilars. There are comparative studies that are required to carry out to demonstrate that the activity and efficacy of the biologic or the biosimilar is similar to that of the original biologic. This is a regulatory requirement also, and it is required for the approval of the biosimilars. It is also used to evaluate the product variant. Say, for example, there is a sequence matching to the biosimilar that is being developed, and the physicochemical properties are also matching. However, if there is a glycosylation difference, the glycosylation differences will highly impact the ADCC activity, especially if galactose afucosylation is there. It has been positively correlated with enhanced ADCC activity. Therefore, it is very important to control and regulate glycosylation at various stages of biosimilar development. It is also important to choose a right candidate. So as we discussed that there could be adverse effect of ADCC, at the same time enhanced activity of ADCC is also recommended. There are efforts being made to select a clone that doesn't have fucose moiety so that we can see increased ADCC activity. And conversely, where the ADCC activity is not expected, different IgG subtypes or mutants are developed where ADCC activity can be reduced. Let's look at
Yeah.
other.
Aditi, there is a
Yeah,
question here.
yeah,
So we have a question from Shalaka, and
yeah.
she's asking like which type of glycosylation enhances ADCC. She has read that afucosylation,
Afucosylation.
yeah, enhances ADCC.
That's correct.
So yeah.
So yes, presence of galactose and afucosylation. Afucosylation is nothing but removal of fucose moiety, brings about increase in the ADCC activity.
Shalaka, is your query resolved? Yeah, Aditi, I think we can proceed. Maybe if
Yeah.
she has anything, she can put it in the chat box.
Yeah, sure. Let me know, Edna. So let us look at different applications of ADCC assay development as well as other activities shown. We have seen that in cancer therapy, ADCC plays extremely important role when there are monoclonal antibodies like rituximab, trastuzumab. They rely majorly on ADCC activity to kill the cancer cell. Also, there are immune checkpoint inhibitors that are ensuring that they block the checkpoint inhibitors, and thus the ADCC can occur naturally by the immune system actively present. Various infectious diseases like viral infections, bacterial infections also demonstrate ADCC activity, and monoclonal antibodies have been used for treatment. Autoimmune disease, where in certain cases like rheumatoid arthritis, ADCC can be used to deplete B cells that contribute to the disease, and as we have seen in case of rituximab, and can be utilized in a way where ADCC enhances and get rid of the cells those can cause autoimmune conditions. Transplantation. So transplantation generally, there is a prevention of rejection of graft, or graft rejection can be reduced or avoided by enhancing the ADCC activity, where the cells that are involved in graft rejection can be eliminated by ADCC. So these are some of the applications of ADCC assay, and can be utilized very well across different functions and treatment modalities. Now let us look at a very typical ADCC assay, how it looks like. This particular assay has been developed at Venture Center. Here on x-axis, we can see the monoclonal antibody concentration, while on the y-axis, we see the effect or the readout of the assay. Potency calculation for the bioassay are always in terms of relative potency, where a sample is always compared with reference to that of reference standard used. Here in this particular bioassay, triplicate samples have been compared at different concentrations of the antibody used. The target cell line that is used is generally the cells that express targeted cancer cell marker, and the effector cell line that has been used in this particular assay is engineered Jurkat cell line expressing NFAT responsive element that drive the expression of firefly luciferase as well as Fc gamma R3A. Therefore, the signals that are coming up, those are coming up from the effector cells. So more amount of the antibody present, higher will be the binding to the target cells, and therefore higher will be the recruitment of the effector cells onto the target cell. And thus we can see that there is increase in the signals with increasing in the concentration of the antibody. Alternatively, previously and in some cases today as well, enriched NK cells are that are isolated from PBMCs are also used for ADCC activity. These NK cells, since they are isolated, they represent the target cell kill, and therefore the readout is of the target cell line. However, this assay brings in lot of variability, because the donor to donor variability would be there, and that at that particular point of time, if the donor has a different immune profile, there will be differences in the NK cells present in the PBMC isolated. There is another specific variant that has been found, which is V variant or F variant. That means at 158 location, it can be either V or F, where V shows higher ADCC activity and F shows the lower ADCC activity. So therefore, depending upon the variants present in the donor, we would be expecting lot of variation in this particular assay. Therefore, it is more these days it is more easily taken up, where there are less variations. Genetically modified cell lines are being used more and more to avoid any inherent variation in the assay. So now let's move to our other mechanism of action, that is CDC, before I talk more on the ADCC assay and the later sites. What is CDC? It is the immune response in which the target cells are lyse through activation and recruitment of the complement cascade to the targeted cell surface. And which are the key components? all different type of complement proteins present shall be responsible to bring about complement dependent cytotoxicity. Let us look at the CDC mechanism of action. We have seen this cancer cell that specifically expresses the antigen. Now, the moment this cancer cell is expressing the antigens in the presence of therapeutic antibody, these antibodies will go and tag to the antigen. Once these antibodies are tagged, the complement C1, that is C1q present in the plasma, will go and bind to this FC portion of the antibodies. The moment there is C1q binding, the other C1, excuse me, the other C1 complex like C1R and C1S also binds to this component, and they form the C1 complex. This C1 complex further recruits variety of complement cascade molecules present, and they bring about the MAC complex, membrane attack complex formation on the plasma membrane of the cancer cell. Now, these complex actually form pores on the plasma membrane through which the cytotoxic or cytolytic components get entry, and thus the cell is killed. So this is about the CDC mechanism of action. Let's look at some of the salient features of CDC. The CDC is tightly regulated process where several therapeutic monoclonal antibodies leverage the power of the membrane attack complex to lyse the cancerous cell. As we have seen that there are variety and n number of complement component present, the CDC, non-specific CDC, has to be highly regulated, and this is regulated at different checkpoints. There are about 50 different fluid and membrane-bound protein components of the complement system that control multiple aspects of physiology and immunity. The complement system is classically associated to a rapid response to invading microorganisms which are either opsonized or directly lysed by the proteolytic cascade of complement proteins as a result of generation of lytic membrane attack complex that we saw in the previous slide. This particular membrane attack complex, or MAC known commonly, there are one copy of C5b, C6, C7, and C8 protein, and up to 18 copies of C9. So this entire set of molecules form one MAC pore that actually leads to the cytotoxic effect on the cell membrane. CDC is effectively triggered by FC domain of IgM and IgG1 monoclonal antibodies, but poorly by IgG2 isotype and not at all by antibodies of IgG4 isotypes. Similar to that of ADCC, CDC is also very well known for various applications, being in cancer therapy, various autoimmune disease, infectious disease, again transplantation, and development of biosimilars. Different comparative studies for CDC are being carried out and are essential to demonstrate that the CDC activity to the original biologic drug is similar to that of the biosimilar in question. Let's look at the CDC assay now. This is the CDC assay, again developed at Venture Center. We have concentration on x-axis and a fluorescence readout on y-axis. As I said, that it is always, the samples are tested always with reference to the reference standard, and therefore the measurement is relative potency. The target cell line shall be the cells that express the targeted cancer cell marker, and complement system used is rabbit serum for the assay. Now, with increasing concentration of the monoclonal antibody, there will be increasing recruitment of the complement cascade, bringing the viability to the lowest level at the highest concentration. Therefore, the readout of the CDC assay is actually target cell line, where the viable amount of the cells are determined. Please note that since it is a rabbit serum that is used as a complement source, there are lot to lot variations that can be seen. Therefore, it is always recommended to evaluate and compare a lot before before exhausting the previous one, because if there are more active complement cascade, then the ratio that you will be adding into your assay will vary, and therefore it is important that if using one lot the assay has been developed, before you shift to or switch to the newer lot, just do one comparative experiment so that the lot to lot variation can be evaluated. So since we are talking about the bioassay, let us see the different challenges and importance of bioassay with respect to ADCC. Bioassays are known to exhibit lot of variability. They are not as simple as that of analytical methods, which are majorly relying on a single equipment. Here, there are multiple variables. Different cell lines are used, different time points are used, different reagents are available, and different incubation times are also there. So the each and every parameter will contribute to the variability in the assay development. So such assays, once developed, they must be repeatable, accurate, precise, linear, covering a wide range of active molecule, and they should also be very robust in nature. ADCC and CDC add a next layer of complexity because there is addition of one extra cell line in case of ADCC and multi-level bioactive molecules in case of CDC. And therefore, the responses given by target cell line. Nevertheless, in vitro ADCC and CDC assays are very sensitive and they are close representation of the in vivo mechanism of action. They correlate very well with the efficacy and potency of the molecule. It is therefore very essential to evaluate the ADCC and CDC activity for a given therapeutic molecule. Additionally, the glycosylation of the MABs are direct indicator of their activity within the system. Hence, if we are looking at a therapeutic biomolecule where we want to evaluate the potencies or activity or efficacy of that molecule for ADCC and CDC, glycosylation of that molecule is also equally important factor to control. So let's see which are the therapeutic MABs that are known to cause ADCC. Note trastuzumab, daratumumab, cetuximab, rituximab, atezolizumab, afatumumab. Note that all of them are IgG1. CDC also rituximab. The primary mechanism of action for rituximab is CDC. Obitunuzumab, ofatumumab. Uh, Also, it's an interesting study where combination of trastuzumab and pertuzumab has been known or shown to bring about more CDC activity. There is another molecule which is ibritumumab, which is a radioimmunotherapy molecule where it binds to CD20 and brings about targeted radiation therapy. And it also brings about the ADCC activity in the cells. Let's look at the other molecules that do not bring about ADCC and CDC. One of the example is adalimumab, as we discussed in the previous slide. Adalimumab, though IgG1 molecule, it is majorly known to bind and sequester the soluble TNF alpha present, and therefore it does not sequester the TNF alpha on the cell membrane majorly, and therefore it will not show any ADCC or CDC activity. Ranibizumab, being a Fab, it does not have a FC portion and therefore shall not demonstrate ADCC or CDC. Other molecules like natalizumab, denosumab, eculizumab, being IgG4 and IgG2 are known to show very less ADCC or CDC activity. Let us once have a look at the different classes of molecules: IgG1, IgG2, IgG3 and IgG4, where we have seen that CDC and ADCC activity is shown majorly by IgG1 and IgG3. We do not see ADCC activity when it is IgG2 or IgG4. So let us revise what we have learned today. ADCC and CDC are two critical mechanisms in the immunotherapy that enhance body's ability to target and eliminate disease cells such as cancer cells or infected cells. Both ADCC and CDC mechanisms are often leveraged in therapeutic antibodies to enhance their efficacy. For example, rituximab and trastuzumab, they are known to show ADCC activity. By engaging multiple immune pathways, these antibodies can achieve a more robust and effective therapeutic responses. There are various ongoing research that aim to optimize the design of therapeutic antibodies to enhance the ADCC and CDC activity. There are other line of therapies where it is seen that no ADCC and CDC activities are demonstrated by the therapeutic antibodies. This includes engineering of antibodies with modified FC region to improve interaction with FC receptor and complement proteins. Additionally, understanding the factors that influence ADCC and CDC, such as antigen density, antibody affinity, effector cell expression levels, also helps in developing more effective immunotherapies. So with this, I would like to conclude my today's presentation, and I'm open for the question answer session. If there are any questions, I'll be very happy to take them. Edna,
Yes.
over to you.
Yes, thanks, Aditi. Thank you very much, for running us through a very simplified version of these assays. These are very complex assays, and their pathways are very. It's difficult to understand, but I think you try to break it down and explain in simpler terms. What we'll do is we'll open the floor for questions. There are a couple of queries in the chat box. So the first question is from Vaishnav, and he says that MABs can be engineered to favor either ADCC or CDC. What structural modifications in the FC region might influence this balance, and how do glycosylation patterns affect their effector functions?
Okay. So first and foremost, there are various mutations that are being evaluated. So FC portion, as I said, that we took one example of eculizumab, where it has been modified in such a way that the FC portion is coming from IgG4 and IgG2, and therefore it overall reduces the binding of effector cells to FC area of the antibody. Also, there are mutations that are being brought up so that there is a reduction in the binding capacity of the FC gamma receptors to respective FC portion of the antibody. There are various antibodies that are being evaluated where the glycosylation of the molecule can be reduced. So as I said, that there can be two scenarios where we want to see additional ADCC activity. There, highly glycosylation, over glycosylation is expected, and therefore it can bring about increase in the ADCC activity. But there are certain cases where ADCC is not expected or CDC is not expected. In such cases, a glycosylation or removal of glycosylated moieties or post-translational modifications can be controlled in such a way that the glycosylation will not happen at the FC portion, thus reducing the binding of antibody to the respective FC gamma receptor.
Okay. Vaishnav, I hope your query has been resolved. Do you have any follow up to this, or should I take the next query, which is by Vaishnav itself? Vaishnav,
Yeah,
if you
hi
could
Edna.
just. Yeah, please.
Yeah, hi Edna. Thank you, Dr. Swati, for this interesting talk. I have a couple of questions, so maybe some 4 or 5 of them. So maybe we can go for the next question and then I can carry on.
Sure. So we'll proceed with the next query, which is regarding the CDC. Is that okay? Yeah. So his question, Aditi, is that CDC depends on density of target antigen and the availability of complement proteins. So are there any major factors that determine the minimum antigen density for CDC activation, and how does this vary across different therapeutic antibodies? So I think it's basically about managing the antigen
Yeah.
density.
Yeah. Vaishnav, to address your question, these studies are still, there are studies still ongoing. It is not very clear as to why certain antibodies show high complement activity and or CDC activity, and why certain antibodies do not show. However, I can tell you one thing for sure, that the complement system is always abundantly present in all circulatory systems that we have. Because CDC is the first line of defense mechanism of our immune system. Therefore, there is no question of what amount or what percent of complement proteins present. They are always there. It is only about the activation of that particular molecule. Another question or another thing to add to your question is that CDC again is also highly dependent on glycosylation of different antibodies that are being evaluated. And therefore, one thing is definitely there, that the amount of antigen present will also impact the activity of the CDC mechanism of action, as well as it also will depend on how specific those cancer antigens are. Because as I said, that if these cancer antigens are present on variety of other normal cells also, then the CDC activity will not be very specific to the cancerous cells.
Sure. I think we'll move to the next question, which is from Abhishek, and then we'll come back to Vaishnav. So Abhishek is asking that ideally for potency-based assays, which replicates should we use, true or pseudo?
Good question. See, ideally, a true replicate is always good to have, but pseudo replicates are also widely acceptable for one reason, because there is a inherent variability present in the bioassays, as we saw, and because there is inherent variability, the dilution or addition of true replicates might bring in more variability in overall assay. And the intention of replicates here is to get the data where you are comparing the reference standard to that of your sample under test, right? So therefore, it is better to have as tight as close data to actually identify a relative potency. So I would not say that a true is good or a false is good. It completely depends on the inherent variability that the assay poses. And pseudo replicates are acceptable. They are not rejected per se by the regulatory authorities.
Sure. Thanks, Aditi. So I hope, Abhishek, your query has been resolved. If anybody else has any more queries, please, you can unmute yourself and you can spend the next 2-3 minutes in, answering that. I have also shared a link for feedback. Please do also give us your feedback so for all the future events, we can then take in all your suggestions and improve recommendations and improve them. So yeah, please do provide us with your feedback as well. Anybody else has any further
Hello,
queries? Yes?
hello.
Yes,
So
Disha.
I have a question.
Yes, Disha, please go
This is
ahead.
regarding C1q binding test. So if I
Okay.
work with a fusion protein, is it relevant to perform C1q assay using HTRF? Because HTRF acceptor is going to bind to the Fab region of the antibody as per the principle. And in case of fusion protein, that could be my functional portion. So is it relevant to perform that test for fusion protein?
See, when we talk, Disha, when we talk about the fusion protein, it is generally the two fabs binding to two different antigens, right? So essentially, there it becomes the antibody binding to these two different antigens are more. So the primary mechanism of action will definitely not be CDC, as far as my
Hmm.
understanding goes, and therefore it is good to show in vitro whether there is any binding seen, because there will be steric hindrance, there will be spatial hindrance that may occur because of two different antigens binding to one antibody. As
Hmm.
far as my understanding and my knowledge goes, the bispecific antibodies are more directed for the anti-proliferative impact or other immune
Hmm.
mechanisms than the C1q mediated cell kill, but I can be wrong also.
Okay, understood, ma'am. Thank you.
Yeah.
Maybe we can take one last, query if
Hi,
any of the
Vaishnav.
participants have. Yes, yes, Vaishnav, please go ahead.
Yeah, hi, Dr. Swati. This is Vaishnav from Intignus Biotech. So, had few queries regarding this ADCC and CDC mechanism. So, the first one is like, since NK cells are the primary mediators of ADCC, and macrophages also participate via YC gamma mediated phagocytosis.
Yeah.
So, do FC gamma polymorphism in macrophages impact the efficacy of ADCC based therapies, or
Yeah. See, Vaishnav, I may not be a very, as subject matter expert for talking about ADCP, because I have not really looked at ADCP so far. But what I can tell you is that the binding, the polymorphism does exist, and therefore the binding also will be different. Uh, NK cells are very, very well known to bring about the ADCC activity in terms of all the cancer therapeutic monoclonal antibodies that we have seen, and very few are known to show phagocytosis during this. However, it is not very clear that whether the phagocytosis is really impacted by I think I'm not very sure about it regarding the polymorphism. Sorry, we can have a discussion around it, but I may not have an exact answer to your query.
all right, ma'am. Thank you so much. There's one more query I had.
Yeah,
So, as you mentioned about an therapeutic antibody like rituximab,
yeah,
which relies on both the ADCC and CDC for tumor
and
cells
also
killing,
apoptosis.
right?
Yeah.
So, do factors like antibody concentration or antigen density or immune cell availability influence the dominance of one mechanism over the other, or is there some synergistic effect of
It is a
both the mechanisms?
synergistic. It is a synergistic effect that happens. See, once you have administered, we do bioassays in very controlled environment, right? You set up for ADCC, only ADCC is going to happen. Same is with CDC, and same with the apoptosis experiments, right? But when the antibody is administered and in vivo, it is exposed to different scenarios, you cannot really predict which precedes over the other. But different antibodies are known to show different preference for their mechanism of action. In this case, rituximab is known to show the primary mechanism of action as CDC. However, it equally shows ADCC as well as apoptosis, but it is not yet very clearly known as to why there will be precedence or why there will be preference of any particular pathway once the antibody is exposed to the antigen in vivo conditions. Yeah.
All right. Can we just go for last question? Sorry.
Yeah, no, absolutely.
All right.
As
So
long
also
as I
some.
can address your question, I will be happy to take them.
No, ma'am, actually, those are very informative. So just wanted to add a bit. So since cancer cells also overexpress some complementary regulatory proteins like CD55 and CD59 to inhibit CDCs.
Yeah.
So are there any strategies by which we can counteract this resistance or enhance the mediated cytotoxicity for immunotherapy especially?
Yeah, there are many. There are many. So let me take you a very nice example, not really example of the CDC, but example of ADCC. You might have known that there are molecules like pembrolizumab or nivolumab. What happens is actually whenever natural killer cells are present, natural killer cells are in their inactive mode because they are engaged by the PD ligands, right? When the PD receptor is engaged with the PD ligand, it will never be active, and it will not bring about the ADCC activity. So these new therapeutic regimens are coming up, and those are showing to be more active and more effective ways of treating cancer, where what they do is that the negative regulators of these cells, natural killer cells, or in your example, the complement inhibitors or checkpoint inhibitors, those are targeted specifically, so that inherent antibodies that are present, that shall bring about the ADCC activity. And also wherever there are inhibitor checkpoints present, and if those are sequestered by the targeted immunotherapy, the classical mechanism of CDC or ADCC shall bring about the cell kill or cancer cell elimination. So yes, yeah, there are a lot of studies going on, and there is entirely new way of thought process where the treatment modalities are changing.
All right, ma'am. Thank you so much for your insightful inputs.
Yeah, I hope I addressed your questions, and I hope you all could,
Definitely, yes,
sail
ma'am.
through the entire presentation, and I did not uh, went into too much of technical. I was told by Edna that the audience is going to be a variety of audience, and therefore I tried to be as simpler as possible and not taking it to the next level of complexity because of the larger audience that I got to know about. So but
Yes.
I'm available. I'll be reachable at Venture Center or individually also for your queries and more discussion where we learn together.
Sure. Yeah, absolutely. So, Aditi, like as I mentioned, that we had a mixed, registrations from the participants, and I think what you have tried to cover today. See, these are very vast topics, and
Yeah.
there is much that you can do in an hour session. But that said, I think with the ease with which you run us through all the details, the applications, I'm sure all of our participants have benefited from it. So thank you very much for taking the time out and for patiently addressing all these queries. And to our participants, also Dr. Aditi is available in case you have any queries. You can write to us, and we'll be happy to facilitate that those interactions. Alternately, we also offer the functional service assays at Venture Centers. You all may reach out to us if you all have any requests or requirements on that front. And lastly, I'd like to wrap the session by thanking you all for attending this session. Please do provide your feedback. I have shared the link there. And this session, as I mentioned, is going to be very soon on our YouTube channel. And the next session will be held in next month. So stay tuned to our website for the details of the upcoming session. So thank you, all of y'all. And with that, we'll now wrap the session. Have a great evening ahead.
Yeah.
Bye.
Thank you very much, Edna, and thank you all the participants. Bye.
Thank you. Bye.